Hey Joe, so I know you're struggling with action potential and neurons. So I'm going to walk through the entire action potential and I'm going to identify what you know well and what's correct and what you're misunderstanding, and hopefully we can help clear up any doubts you have. So let's start with a picture of a nerve cell, a neuron. So you'd first said let's start with the first step of the action potential. So you'd first said that the dendrites shoot out neurotransmitters. So that is not necessarily correct in regards to the initial process. So neurotransmitters like glutamate will bind to dendritic spines. Then the signal will travel down the dendrites to the soma or cell body, which you had correct. And the soma and cell body, specifically right on this area, the axon hillock, that is when the signal is added up. And once the signal is added up, if there is a voltage change, then the signal will fire, kind of like a gun. It either fires or does not, depending on the change of the environment in regards to voltage. So if there is enough of a change, the signal will fire along the axon, which is the structure I am tracing here for you. Now the axon, covered in myelin, which speeds up the signal. This is the most important part in terms of the ion channels, in regards to the polarization, depolarization, and those kinds of things. So let's examine that a little further. So the signal fires along the axon. So at these spaces called the nodes of Ranvier, I'm going to zoom in. There's kind of a zoomed in drawing of it. You have these ion channels. You have sodium, potassium. You have a sodium-potassium pump. And so what happens first, and this is kind of where you get confused in the order. It is the sodium channels, not potassium protein channels. It is the sodium channels that will diffuse, you had that correct, diffuse their ions from outside to inside. So normally the resting potential between the outside and inside in regards to the ions is negative 70 millivolts. Now when the voltage changes and reaches a threshold between negative 50 and negative 55 millivolts, that is when the sodium channels will diffuse their ions, because that's when the action potential has been activated and set on the axon. So the sodium channels will open up all along the axon first, and then once they have opened up and then finally closed, then what's going to happen is the inside space, which was more negative initially, and the outside space that was more positive, that's going to begin to change. The inside becomes more positive, the outside becomes slightly more negative, and so what's going to happen is then the potassium channels are going to open up, and so they're going to do the same thing, open up all along the axon, and the potassium channels want to reestablish the more negative inside, and they want the outside to be more positive, and so potassium will be pumped out. So the first step in that process is depolarization, because you have less of a disparity between the inside and the outside in regards to the voltage or the charge. That's depolarization. So if you looked at a graph, it would go up, that's depolarization, and then it's going to reach a maximum. So right around 30 millivolts, that is when it's reached its maximum, and then you want a negative charge to be reestablished. So then you've reached that maximum, and then you're going to come back down. So it looks like a bell curve. So that down portion of that bell curve after you reach the maximum, that is called a repolarization. And so you're reestablishing that the inside is going to be more negative than the outside. And so the repolarization occurs. But what happens is it goes a little too far past negative 70. It becomes way too negative. And so to reestablish the correct balance in terms of the resting potential, the sodium-potassium pump kicks in to help reestablish that negative 70 millivolts. So that is what's happening along these ion channels here at the nodes of Ranvier. Now we get to the end. So the axon terminal and the terminal buttons. So you can kind of zoom in on it. So you have these vesicles with neurotransmitters, and that signal is traveled all the way down. And so what's going to happen is that signal is going to cause these calcium ions to flow and these calcium channels to open up, and they're going to cause the vesicles to bind to the cell membrane, and they're going to emit the neurotransmitters via exocytosis. So everything in this process, to kind of summarize, just think about it as kind of like a chain reaction. Everything wants to be in equilibrium, and everything has a certain step in order to reach equilibrium and affect the next. So with that in mind, I hope I answered all your questions, and try and remember these steps and draw it out, because that will definitely help you in terms of remembering the order with this specific function. So thanks.